So today we're going to start with another extraordinary guest, Professor Francesco Di Pierro, biologist, pharmacologist, immunologist. He deals with phytotherapy and bacterial therapies. He's always busy, so I figured out how to bring him here. I think, according to me, he's excellent, and he will talk about bacterial therapy. He will tell us something that you will hardly hear around. Can I have your attention, please? Okay, thank you. Dr. Di Piero, I give you the floor and follow him with attention, please. He speaks very fast, but I ask him to understand well what you're saying. An applause for Dr. Di Piero. Thank you. So, okay, thank you, Dottoressa Di Modugno, for these words. I will try to deserve these words. Theme of the day is bacterial therapy, which aims at use bacteria as a therapy that is not so innovative. Maybe you can say in a pharmacist they sell probiotics, so I've already heard about it. No, it's not like that. You can think about using bacteria, exploiting its killing capacities. So bacteria kill each other every day. So a bacterium living in an intestine of a person and living in an oral cavity or in a vagina is not easy, because bacteria are thousands and thousands and are not like us. We're comfortably seated. They're overcrowded and they're inert. The bacteria are like are no dormant, and they have to defend themselves every day. So living for a bacterium is not like living on a culture dish. That's easy. We feed bacteria that way, but it's a passive life. But everyday life, not in a culture dish, it's an active life. They have to attack and defense. And there are some aggressive bacteria. But they're not bad for the host body. So I can have aggressive bacteria, but they are not able they're not pathogen. They're bad only against 2 or 3 bacteria, not towards everyone. It's like a metaphor. So we invent. It's I'm against Portuguese people. It's a metaphor. Portuguese become a problem, and I'm useful in a colony because I attack only the Portuguese people. So this way we can think to prevent and use preventive medicine. And we use antibiotics not in a preventive way, but we can use bacteria for this reason. So this is bacterial therapy. This is what I'm going to talk to you about. And they exist to kill each other, and they kill through poisons. These poisons belong to antibiotics class. Everyone knows what antibiotics are, but these non-bad bacteria don't use antibiotics. They use a molecular class, a well-known but never exploited, the one of bacteriocins. The microbiology knows these types of bacteriocins, but they're not well implemented. They're not well used. bactericin attack everything. The antibiotics attack everyone, while bactericin don't do that. So it's useful to know some weapons that can attack certain bacterial species. This is the theme of the day. And we are going to differentiate what antibiotics and bactericins are and how can we can use them. Starting from this image, this is a work in one of the most important journal, Nature. Nature is really famous. And a few months ago, Nature published something similar to what I'm telling you. They study from the assumption that patients, some pathogens like in nose, in throat, like Staphylococcus aureus, are not particularly pathogen and they're not antibiotic resistant. But in the hospital, if there are these Staphylococcus in your nose, there's a risk of spreading an epidemic inside the hospital. So this study underlines something different. They noticed that some patients in the hospital didn't have Staphylococcus aureus in their nostrils. They have Staphylococcus longdunensis. We don't have pathology caused by this Staphylococcus, but these hospitalized patients didn't have Staphylococcus aureus in their nostrils. But why? Because if we take lugdunensis in these and we put it in a culture dish where we're growing Staphylococcus aureus, which is antibiotic resistant, it will grow, lugdunensis will grow. Can you see these death zone? This white part is the Staphylococcus aureus, one of the two or three that risk to cause epidemics that can kill us. So it was enough to put this bacterium on a culture dish, and all around the Staphylococcus aureus died, so was killed. Today we have no drugs to cure this aloe. And this bacterium killed the other one. So going forward, they discovered that subjects, lugdunensis positive patients, secreting a poison called lugdunin, which is lethal for Staphylococcus aureus. So this patient didn't suffer from any disease. So the risk of dying for Staphylococcus aureus infection is 0.059. The risk of dying if you don't have lugdunensis is 0.347. So eightfold more. So everyone is 8 times protected because there is a particular bacterium inside us. So when a subject is hospitalized and is at risk of infection, we do a nasal seeding of this bacterium because this bacterium will deal with the problem, will secrete poison against aureus only if aureus will occur. So these researchers concluded their work saying that our studies suggest that the probiotics concept, which deal with intestine, the concept should be extended to body sites other than the gut, such as the nasal mucus. And they studied the competitions in a nose. Competition not for a cold, but to save a life. And it's the first one in the world who show to the scientific community that it is possible to exploit bacterial competition. They kill each other because they attack each other or defend. Who discovered that? John Tagg. One day he decided and he thought about the situation because some kids got infected really frequently because of Streptococcus pyogenes. They get infection in their throat. So Streptococcus pyogenes in the pharyngotonsillar zone. So how come these kids get sick 8 times in a year? Some they will never be sick, but why? The modern medicine would say differences are in the immunitary system. I'm an immunologist, and I say that this is something foolish in reality, but some kids were born with a throat which is rich in bacteria, and this bacteria can defeat other bacteria. So he asked to the government to carry out some tests, a test in particular, so an oral swab to 780 kids. Then he took a culture dish, and in half of the dish, he put a seeding of these swabs. And he said, according to me, I will see that some kids, some throats, will attack the streptococcus. Others won't do that. And he noticed something like that. He noticed that there were some kids using an algorithm 677, and these throats were very aggressive. There were intermediate kids, so 2 to 6, and they had less bacteria, and kids 000, where the pyogenes would attack the swab, and there was only streptococcus in these culture. He did a prediction. Since I pinpointed these 3 categories, the kids, the 677 kids, in comparison to the others, will get less sick and will suffer less. So he obtained the permission to follow these kids, and this is the first demonstration that having bacterial flora in a particular part of the body is beneficial. So these kids would get sick 50% less. So this is great. What the nature researchers said last year, TAG said that in 1999. But why did he choose to say 677? Well, he carried out a study in 1978, and he understood that if I put two bacterium one against each other, they fight. So he make them fight, and he understands that some bacteria are more characterized than other. This is a metaphor. If I were blind and I wouldn't know, and I didn't know how to identify a muscle, I would say humans are fat, slim. Doctors, you have muscle, so you fight first with the superhero, then with the gym obsessed, then with the kid. On the basis of the winning, I decide the results. There are millions of bacteria. Then I try to make one bacterium fight with the other's bacteria. So the idea would be finding a bacterium which has 9 pluses, so able to kill all 9 bacteria. and plus, it doesn't. It's not pathogen for us. This is a dish which is not very cannot be easily understood. The P5 is killing the 9 indicators, so 9 types of a person, 9 streptococcus indicators, which can cause infections and diseases. This bacterium kills them all, and an algorithm was developed, the 777, identifying a bacterium killing all the lookalikes. In this work, he found 32 bacteria killing the others. But why didn't do anything? Because they were pathogens. According to Langsil classification, these are a serum, which is pathogen for humans. So he couldn't use these because they were bad bacteria. But the idea was right. The idea was right. Find something non-pathogen that is 777, and he managed to find it in a first grader. He found a throat capable of attack the pyogenes. And this was the Streptococcus salivarius. So 80% of our mouths is full of this bacterium, which is not pathogen. So it's a simple bacterium. In this kid, one particular salivarius called K12, because it was the number of the kid, it was a 777. So we found a non-pathogen killing all types of pathogen pyogenes in man. But why? Because he produced 2 poisons, which have curious names, salivaricin, and they are identified as A2 and B. These 2 poisons produced by this salivarius kill all pyogenes in the world.
L'unità di tempo. Questa è una piastra dove ho strisciato lo Streptococcus pyogenes e questa patina bianca che cresce. Poi dall'alto faccio cadere una goccia di alcol. Aspetto mezz'ora e vedo cosa succede. L'alcol uccide, disinfetta. Bene, l'alcol...
Alcohol produces a hole. I let betadine flow also, and this is another hole. Then I use other substances. When there is the K12, in half an hour we see cells of K12 growing, spreading poison all around and killing. This may mean that the K12 is particularly harmful. When there is a lot to eat, he eats just faster than other. So it proves that it's starving, so it eats a lot. There is a killing test that is delayed over time, and so I do that. I get a clean dish with K12 coming from this kit vertically. I make it grow for a while, for a few days, then I kill it. So there is no more, it's no more there. Then I get the pathogen pyogenes for the man, and I make them grow horizontally, but they don't grow where the K12 was because they are not able to grow there. He's dead, the K12 is dead, but he left some poison behind, so it's not possible to grow anymore there. The poison don't allow other pyogenes to live there. So you might get the idea now that we can get some people that are always sick because of Streptococcus in throat, and so we give the K12 strain so as to have a colonization, and so we can see if there is a real protection from the Streptococcus. It was done in 2012 for the first time in Italy. this is one of the most important articles with 40 adults were tested with Streptococcus infection at least 4 times per year. So that means they had to take 4 times antibiotic during the year, 4 or more times. So there were 2 groups. One group were given the usual treatment, and the other case, they were given the K12 strain. What happened is that the second group got sick 83.7 less time than the other group, just because of Streptococcus. After a few months, while administering this K12 strain, they interrupted the administration so as to see if the protection and the follow-up during the follow-up would go on without actually administering the medicine. In the 6-month follow-up, they had been protected 62% more than the non-tested group. I showed these results in the hospital of Turin, so I suggested to make the test also for kids. About 100 kids in a group of 41 kids getting sick very often because of Streptococcus. They were given this strain, and they got sick 92% less than the other group, than the normal group. Here, when there was a follow-up period, that was difficult to get approval from the parents, because they were really satisfied with these results. However, when the administration was stopped, the protection was 62%. It's not just me saying that the bactericins are effective. This is medical literature. For example, this is an article from 2014, Frontiers in Microbiology, talking about the antibacterial activities of bactericins. Talking about the development in foods, the application in foods. For example, we can use this in cold cuts or as pharmaceuticals. So this is not a thing I'm just telling you, but this is reality. It's just not easy to find them. John Tag took 30 years to find the perfect strain, because we have to decide whether they are hostile towards the host. This is a lab test. For example, you see that one doesn't have the poison. Here is more interesting. Do we have this kind of bacteria? This is a culture dish of a 6-month-old breastfed baby. And we are analyzing Enterococcus. Here we see a larger image of a specific area. This is a bacteria colony, and these are 3 bigger ones. Around them, there is nothing, because they are killing the similar bacteria. So we may decide to take this bacteria, but it makes no point, because they are killing non-pathogenic bacteria. So they have to kill few bacteria, not being pathogenic, and their poison should be aimed at the pathogenic agents. This mechanism, however, we can see that it's really common. You see around the dish that there are similar bacteria. Here we get closer. so we can understand how the killing process takes place. This is an image from 2011, and this is Streptococcus agalactiae, which can be found in the vaginal area. If the woman who is about to give birth has this kind of bacteria, this is harmful for the baby. If the woman is positive for this kind of bacteria, there is an antibiotic therapy, but the baby is already taking antibiotics himself. So he's losing precious bacteria. So if we take bacteriocins that are lethal for Agalactiae, we make them drop on the culture dish, and we wait 180 seconds. We see that 180 seconds after, some of the bacteria exploded, popped, and all the material inside is flowing out. In a while, they will all die because bacteriocins are making holes in the cell. These are other examples for this kind of holes. These are salivary 9, and this proves that there are many cases. The difference between the picture A and B is 30 seconds. You can see in the second picture that there is a hole in the membrane, and in this case, it bursted. There is no material anymore, and you see that just a little piece of bacteria that is still attached. What I showed you is important for many experts, and this will sure remind you of antibiotics. And you may think that these two elements are not so different. So let's analyze what the differences between antibiotics and bacteriocins are. Why are antibiotics good, seen as good, and bacteriocins as just as good, but they kill less? Why is there such difference? I listed their general characteristics. Antibiotics are secondary metabolites. That means just as bacteriocins. So there is no difference in that. Antibiotics have a protect structure, but also non-protect structure, while bacteriocins are all with protect structure, protein structure. Antibiotics and bacteriocin both have quorum sensing. That is that they understand each other, they communicate to each other, with each other. Not through senses, but they manage to attack our host when they are in a determinate number. And they do just that when they know they are able to do so. Then this is what quorum sensing is. So that they perceive how many they are. They count themselves. And this is characteristics for both categories. They have an antibiotic role, such as bacteriocins. And here is the differences. Antibiotics have wide spectrum, and bacteriocin narrow spectrum. So antibiotics attack thousands, while bacteriocins are targeted to a specific bacteria. This allows me to say the roles in evolutionary terms, which is kind of interesting. First, we have to think that the first guests, so to say, in our planet arrived 3 billion years ago. But 650 million years ago, there was still nothing, because the creation of the word, let's say the living word, is 600 million years ago. Before, there was just rock and just a little water. But we already had bacteria. We didn't have just the environment. We have one big environment. It was just a unique and only environment that was inhabited by all bacteria. And then different environments developed. I'm not talking about water and rocks, but also, for example, a vagina of a carp or of squirrels. It's tobacco, it's leaf, or maple leaf. It's different environments for the bacteria. So they are inhabited by different bacteria. So as a bacteria, I will keep on living in the perfect environment for my survival. And the different environments will never get in touch with each other. So there is a specific molecule for the specific environment. That means that antibiotics have developed themselves into bacteriocins. Before, antibiotics were active on the unique environment that was available, and then they developed themselves into bacteriocins. So this is why it's needed, because the bacteriocins are helpful to target a specific environment. And so bacteriocins are much more specialized. Another difference is that in the case of bacteriocins, the structure is just and only protein structure. And through this, I can find out that there is a similes in the phenomenon of bacter conjugation, that is, so to say, the sexual function of bacteriocins. And this is helpful to mix the DNAs. This is what happens also in parents. So they rearrange the DNA, they exchange tiny pieces of DNA, and so they may be stronger to environmental changes. And this reminds us of bacteriocins from a chemical point of view, because bacteriocins have evolved from sexual function elements. I would like to talk to you about quorum sensing. This is a decision that the bacteria take to decide whether to do something or not to do something together. This is, for example, a throat of a kid. If I make a swab, I will find this bacteria. That doesn't mean it's necessarily infected. For example, also in women who have the bacteria for thrush. If the bacteria are not able to expand themselves, they will never take the decision to attack. How do they know whether to attack or not? They secrete bacteriocins, and through bacteriocins, they can communicate to each other, understand how many of them there are there. So if there's many of us, for example, we can all attack. And so they transform colonization into infection. What changes is the virus aspect. So the force they have, they don't waste energy if there are just few of them. But if we are together, we can attack as a whole.
But how are these bacteriocins classified? The most studied one is the first class, so the gram-positive bacteria, like antibiotics, which is quite funny because who studied bacteriocins was like envy and envious about antibiotics. But they're not, they're quite different. These are a group of bacteriocins characterized by an amino acid, lanthinin. So bacteriocins containing lanthinin are called lantibiotics because they have lanthinin. Lanthinin looks for a molecule called lipid 2, and when it finds it, it makes a hole. So here the evolutions related to the presence of lanthinin. This bacteriocin finds the lipid 2, they combine, they polymerize, and make a hole. And from this hole, cytoplasm will come out, water will come in, and the bacterium will explode. So they will die. Obviously, this works because bacteriocin takes time to find lipid 2 and make the hole. But how? It has positive charges, and for electrostatic force, the bacteriocin can work this way. Obviously, there are a lot of bacteriocins. I showed you some photos, but there are bacteriocins making no holes, and they prevent other bacteria from doing resistant walls. So they can inhibit the wall membrane. Nature published that the Streptococcus salivarius we were talking about, one of these poisons, the salivarisin B, has this behavior. So it inhibits the creation of the wall. One strain, how many bacteriocin can a strain produce? I gave you an example of the salivarisin 2, but this means that all strains produce one bacteriocin? No. Some strain produce one bacteriocin, the others more bacteriocins. For example, other Streptococcus salivarius, a lot of them produce salivarisin. Some produce 2 bacteriocins. Some even 3 bacteriocins sometimes. It is important to prefer a producing strain which produce a lot of bacteriocins or the other way around. I'll give you an example. This author found Enterococcus faecium T136, and he says this strain produces 2 enterocins, A and B. But what's the interesting fact? The strain, so they were cultivated. One is killing the bacteria, the other one is not. So this is similar in every species. But where can we find DNA pieces in these bacteria? On the wall, in the cytoplasm. They have 2 possible locations. Plasmidic localization, so outside the chromosome, and the chromosome localization. I'll give you another example. So the 18 strain, N18, Some of them are a chromosome location, others are megaplasmid location, and useful to prevent tooth decay. And it's used in those kids suffering often from tooth decay. So it kills the mutants because it produces 4 poisons. One, salivary M, starting from a chromosome. And it starts from a plasmid, so it has both possibilities, both locations. But the natural question is why the strain, a bacteriocin doesn't kill the producing strain? Because the immunity factor exists. I'll give you another example with another enterocin. This is a bacteriocin called the pediocin. And it has a strange form, shape, and it tries to separate these walls. When it does, it creates a hole. But why this wall? Why this wall is not? Why the wall of the bacteriocin are not destroyed? Because it has immunity factor. So imagine they're running in the cytoplasm, they climb, and the poison will come out eventually from the bacterium. And the immunitary factor will stop in the membrane of the bacterium. When the poison is out, the poison will try to open the wall of the bacteriocin, but that won't work. The immunity factor is faster and will bridge the gap in the wall of the membrane. So bacteriocin producing bacteria,
with some exceptions,
don't die because of their poison, because they have immunity. So if I were you, I would be really curious and I would say, so this means that there are bacteriocins with weapons and those who haven't weapons. Those with weapons are aggressive. But why don't we all have weapons? Bacteria should be all like that. There are 3 types of bacteria. The bacteriocin-producing bacteria. Then I'll find the resistant bacteria. They lost poison, but they kept immunity. And then I'll find sensitive bacteria, because they die because of bacteriocin. But this is a disadvantage. In order to explain to you that, I will show you what happens. When a colony is not disturbed by an enemy, is not compromised, the producer, the immune producer, has immunity and the bacteriocin gene. Then some bacteria lost bacteriocin, but they kept immunity. Other bacteria lost everything. So no war. There's no war inside, no way to attack or defense. But if I don't have weapons, it's really hard for me to combat, to attack. So I'm a loser. I'm in difficult. But if in case I have pathology, I'm protected. But why do we have these? Do we have these 3 situations? there's no win situation because scissors win paper, but paper loses with the stone
because it's more expensive.
because it's more expensive.
And this means that if we have no bacteria before turbines,
So in a colony, in a bacterial colony, I can have 25% of PI strains, 50% of sensitive strains. And they proved this was right. If there's a war, things are different. We become immune producers because some of them tend to transfer plasmid to sensitive subjects. So it extends, the colony extends. The last
question is, are bacteriocins resistant? Because I don't want to tell you to stop using antibiotics, but to use them in a better way, because they induce antibiotic resistance. That means we have to find ourselves in a situation where we can use them less. So we have to analyze if bacteriocins are just as effective. But remember, it takes 20 years to find a poison, and in the world we can find in 9 months the first antibiotic resistance, so it's a no-win situation. In fact, you see the authors say there is concern because there will be also bacteriocin resistance, maybe. There is just one bacteriocin in the world that is used without the bacterium. It's called nisin. And this has caused some resistance. If we use bacteriocin just as antibiotic, over time, the bacterial evolution will produce resistances also towards bacteriocins. Remember, the man didn't create the antibiotics, but these were created by fungus and bacteria. The man created the antibiotic resistance. We didn't have antibiotic resistance before we created it with the wrong use of antibiotics. There is no resistance to bacteriocins, but if we use them as antibiotics, this will take place as well. So the best strategy, the winning strategy, is not using bacteriocins as antibiotics. We just call them in a different way, but using the strains producing bacteriocins so that they can decide whether to produce them or not. This way we won't develop resistance. So we can use bacteriocin-producing strains, but it's better if we don't use the purified bacteriocins, because that means having new antibiotics, basically. Do we have any proof of the non-production of resistance? Yes, always talking about the K12 strain. There is still no resistance that is linked to the K12 bacteriocins. The bacteriocins, and there is no resistance against the bacteriocins produced by orally administered traits. At the end, so we have the concept, the idea of bacteriocin therapy. So we have to adapt ourselves to the germ warfare. So the fact that they kill each other, but we have to use bacteria to prevent infection. Why should we prevent? Antibiotics to be effective doesn't need colonization. Bacteriocins, if I just administer them, I can kill bacteria. But if I do so, I create a society, so to say, where the community is drowned or inside bacteriocins or antibiotics, and this is what produces resistance. If I use bacteriocin producer strains, they produce it after the colonization. So they can proliferate, and if there's need, they will produce also the bacteriocin. This way, this can be used just as a prophylaxis and not as a therapy. If I use the K12, there is no possibility, if there is already an infection, there is no possibility for colonization because there's no space anymore. Otherwise, if, for example, the tonsils are clean and not infected, the K12 can colonize for example, tonsils. And so this is an advantage because it can be used as a prophylaxis. So as a sort of vaccine, so to say. But there are difficulties, as you can understand, for the therapy use. So we can use the bacteriocin-producing strains as a prophylaxis, as a prevention. And these are in patients where there are recurrences in infections, in specific infections. For example, through streptococcus, and this may reduce the infection as well as the antibiotics use. Is that really so? Yes, there is a research that was carried out in Milan with an analysis always of the K12 strain, 30 families. A group was told to use just antibiotics, and another group was asked to use the K12 strain. And this is differences. 30 families using K12 used as a whole 30 dosage of antibiotics, while those using the K12 before used 900 doses. So there was a reduction of 34th time. Thank you.
Grazie, grazie. Un bellissimo applauso.
So thank you very much. So please clap your hands for this wonderful speaker. He is a doctor, he is a professor, so you see him, he looks like a child actually, but actually is 50 years old. So I must say this because we are talking about getting younger. So and this is a real strong man. So he looks like a child, but he's not. Thank you very much. So he is very humble, but I really like him.
Grazie ancora Francesco.